Hello and welcome to the nervous tissue chapter, chapter 12. This will be one of 5 nervous system chapters we will be studying this semester. So, quick introduction. So we'll start off by talking about how the nervous system is laid out. We have 2 main divisions, the central nervous system, or CNS, and peripheral nervous system, or PNS. As you can see, the central nervous system is only consisting of the brain and spinal cord, whereas the peripheral, we have cranial nerves, spinal nerves, ganglia, enteric plexuses in the small intestines, and sensory receptors in the skin. To further break it down, we see the peripheral nervous system is broken into 3 different parts. We have the somatic nervous system, which is going to control skeletal muscles, right here. The autonomic nervous system, this is where we think of our automatic responses, our involuntary responses. This is going to control smooth muscle, cardiac muscle, and glands. We see the sympathetic and parasympathetic divisions as well in the autonomic nervous system. This is your fight or flight with sympathetic, or rest and digest with parasympathetic. In the GI tract, we will see the enteric nervous system. This is going to be controlling smooth muscle contractions, gland secretions, and endocrine cells within the GI tract. Okay, so a quick look. The brain, housing 85 billion neurons. The spinal cord, 100 million neurons. So functions of the CNS, we are processing sensory data, so all the sensory input we gather from our body, we will be processing that within the spinal cord and brain. We will initiate motor commands, so in other words, telling the muscles and organs what to do. And memory, emotions, and learning will all take place in the brain. In the peripheral nervous system, it consists of all nervous tissue outside of the CNS. So nerves, these are bundles of axons, ganglia, small masses of neuron cell bodies. We have enteric plexus, again located in the GI tract, and sensory receptors. So we're monitoring everything from internal and external environment with pain receptors, touch, vibration, temperature, and so on. Okay, so taking a further look at somatic nervous system, we have receptors pretty much everywhere. So head, body wall, and limbs, where special senses of vision, hearing, taste, and smell. Then we've got motor neurons that conduct impulses from the central nervous system to skeletal muscle only. So motor output is only through skeletal muscle. The autonomic nervous system is going to be sensory neurons pretty much everywhere as well. Mostly in the visceral organs, so we think of the stomach, the lungs, the intestines, where motor neurons are going to be impulses from the CNS to smooth muscle, cardiac muscle, and glands, all involuntary situations. And then 2 divisions of the autonomic will be sympathetic, or fight or flight, and parasympathetic, rest and digest. Enteric nervous system, also known as brain of the gut. As you can see, over 100 million neurons. So we have just about as much in the enteric nervous system as we do the spinal cord. A lot happening here. So sensory input through the gastrointestinal wall. Motor output through the GI tract with secretions and smooth muscle contractions. Functionally, the nervous system is divided into sensory, integrative, and motor. So sensory, this is where we are sensing the different changes in the body through those sensory receptors. Integrative, we are analyzing that information and making decisions on what to do with those. And motor are our responses to those decisions. So the two different types of tissue, we have neurons and neuroglia. Neurons are going to be the functional units. These are the units that are electrically excitable. They are sending the nerve impulse or action potential. Structure of a neuron, we have dendrites, cell body, the initial segment or axon helix, the axon itself, and telodendria or the axon terminals. So this is going to be the structure of a very common multipolar neuron. The dendrites are all these projections off of the cell body. We are receiving information through these. The cell body itself, seen here with all the organelles and the nucleus. The axon helix is going to be the beginning of the axon off of the cell body. Then we have the axon itself. We only have one axon for each neuron. As you can see, we can have multiple dendrites, but only one axon. We can have branches off of that axon, but from the cell body itself, there is only one. And then the axon terminals are going to be seen at the very end, and those synaptic end bulbs will be the attachments to the next cell. Okay, so this is another look. So the synapse. The presynaptic cell will be the neuron sending the message. Postsynaptic will be receiving the message. The synaptic cleft will be the gap between these two connections. Structurally classified neurons, the majority of what we see are multipolar. So most in the CNS and then all motor neurons. Bipolar, we are specialized neurons for the retina in the eye, the inner ear, and olfactory area. We will see these when we get to special senses. And then unipolar. These are going to be sensory receptors. You can see touch, pain, pressure, and thermal or temperature. A couple other examples, Purkinje cells, very specific, and pyramidal cells. So they look like a pyramid. Okay, so looking at another way of labeling these neurons, sensory neurons are also known as afferent. So we are picking up information. We're monitoring the environment within and without. Interneurons, these are between sensory and motor neurons. We are distributing that sensory information and deciding where to act on a motor neuron. Motor neurons, also known as efferent neurons, we are carrying out the instructions from the central nervous system. So some quick examples of sensory receptors. We have corpuscle touch, obviously for touch reception. Laminated corpuscle, more touch. Then mechanoreceptors for mechanical sensations, so movement, touch of the skin as well. Nociceptors are pain receptors. Neuroglia, these are the cells that are holding the nervous system together. So they are not electrically excitable. They do not send signals to anywhere. They are making up about half the volume of the nervous system, and luckily they can multiply and divide. We have 6 kinds. We have 4 in the CNS and 2 in the PNS. So the 4 in the central nervous system, astrocytes, oligodendrocytes, microglia, ependymal cells. 2 in the PNS, we have Schwann and satellite cells. So their main functions will be seen here. Astrocytes, blood-brain barrier, deciding what material can make it through to the nervous tissue of the brain. Structural support, and we regulate functions. Oligodendrocytes, we are forming and maintaining the myelin sheath around axons in the central nervous system only. So we will see how that creates an insulation and faster production of those action potentials. Microglia, they are kind of the immune cell of the nervous system, or the central nervous system. They act as phagocytes, cleaning up debris. Ependymal cells, very specific. I did mistype this. This should be CSF. It is cerebral spinal fluid. So we are producing, monitoring, and assisting in the circulation of cerebral spinal fluid. Schwann cells, we produce and maintain the myelin sheath in the peripheral nervous system axons. And here we can actually help repair damaged neurons as well. Satellite cells, we surround cell bodies in the ganglia of the peripheral nervous system for protection and regulating functions as well. Here's a look at all of these. Astrocytes. So they're named. Astro is star-like. Cyte is cell, so a star-like cell. Microglia, a little smaller. So these oligodendrocytes, you can see they have multiple projections, creating myelin sheath around 2 or more axons, or at least 2 or more places on a single axon. And then ependymal cells, they create a sheet of cells for filtering blood to create cerebral spinal fluid. In the peripheral nervous system, the Schwann cells, we have a single Schwann cell creating a myelin sheath around a single area of an axon, unlike the oligodendrocytes. We do not have to produce a myelin sheath. This would allow us to surround multiple axons for unmyelinated axons. Satellite cells, you can see they are surrounding the cell bodies. This is for protection and regulation. How we produce that myelin sheath: a Schwann cell comes in contact with an axon. We slowly start to wrap around that axon within the cell. Speaking of myelin, this is how we get the difference between gray and white matter. So white matter is mostly consists of myelinated axons. Gray matter is going to be mostly cell bodies and unmyelinated axons. So you can see white matter in the spinal cord is more on the peripheral, and then more internal in the brain. This makes for quicker connections from one area of the brain to another. So we can travel in a straight line instead of connecting and traveling a farther distance.